Yeah, thanks very much for the invite. It's exciting to talk to you about this work. Um, I'm going to do my acknowledgement slides first to make sure I don't forget folks. This is really just as many of you know that are on the call or here, this is a long time, what I'm going to talk about today is a long time collaboration between my lab and Kelly McNagny's lab in the Biomedical Research Center out on campus. And the folks that are really doing the work at the moment are Aaron Bell and Pam Dean in my lab. But you'll hear some stuff that Marcia Graves once did in my lab together with Julie Nielsen. Julie's actually now working in Brad Nelson's lab in Victoria. And the original ovarian work was done by Jane Cipollone. And Arun Somasari, the guy that runs Waxit, actually did the first Podocalyxin in cancer experiments. And then in Kelly's lab, Julianne Brassard, who many of you know because she's part of the new TFRI project in endometrial cancer that I'll briefly allude to, and Mike Hughes are running things. Diana Canals actually did a lot of the antibody and antibody drug conjugate work I'm going to talk about. Kim Snyder did a lot of the original functional work with Podo. And Julie, together with Marcia, actually showed that podo does weird things to the edges of cells, essentially generates protrusions. And we think that's how part of the reason why Podocalyxin helps drive invasion. I was putting this talk together, I realized, probably many of you on the Zoom, how many folks we've roped into this little cottage industry over the last few years. There were a number of folks originally at the Center for Drug Research and Development, now Admera, out on campus, who have now gone off to other places. But John Babcock, Chris Bond, and Ishmael actually generated all the antibodies I'm going to tell you about today against this molecule, Podocalyxin. A fellow named Ola Blix in Copenhagen and his student Iliara have helped us try and figure out what the antibodies are binding. And then various clinical translational collaborators. I just have David and Steve here from Pancreas BC, and many of you, of course, know Steve Calliger. And we've also started to look in pancreatic cancer as well. But this all started with David and Blake, who allowed us to stain TMAs, the original breast and original ovarian TMAs at GPEC. And Martin Coble, when he was a resident here, really helped us out to figure out what was going on. And more recently, Samuel Leon. And I didn't know where to put Brad. I should have had a different column for his group over in Victoria, but he is part of OCARE as well. And Brad's helped Julianne with some of the tumor infiltrating lymphocyte stuff. And then now, with the new Terry Fox group grant that Jessica put together, and Yemen is really running one of the projects, Project 3 in that grant, together with Don helping us out, and then Navina Lynn. And Spencer's done a lot to help us look at where Podocalyxin is in the female reproductive tract. And we're doing a little bit of work with Mike as well, treatment, post-treatment stuff. All right. With my acknowledgements, I could stop. No, I guess I should go on. Yes, Yemen. Okay. All right. This is the reason for the title. And I'll tell you about Podocalyxin for those that don't know about it in a second. This foot-like, this molecule that shows up in the foot-like processes in kidney cells, in podocytes. But when we originally screened with an antibody from a collaborator named David Kershaw against Podocalyxin, which is a cell surface molecule, one of the interesting things in the GPEC ovarian array was in high-grade serous tumors, essentially 90% were positive. And about a third of those, this was what really Martin helped us work this out, was clearly on the surface. Sometimes there was a cytoplasmic staining, although it's turned out that there was also some surface staining there as well, which is why there's been some differences in what we thought. There is a little, and this is just showing you what that looks like. So just immunohistochemistry, and what we're staining for on the HTSOC is just the Podocalyxin in brown, and this is the surface stuff. And it's really most often when you have polarized apical basal polarity in the cells, it's very often on the apical domain. And that's the way it is in normal cells. And then this is the situation where what Martin scored is cytoplasmic, but it turns out there's some membran staining there as well. We figured out. But when we originally looked at the array and scored it membranous versus cytoplasmic, there was some prognostic significance, not huge, and that's gone away now that we have different antibodies that Sam Leong helped us with the scoring. Point is, Podo's on tons of high-grade serous tumors. It turns out it's on lots of, quite highly expressed on lots of others. Originally, with David and Karen Gelman in the original breast TMA, we had seen a similar thing in invasive ductal breast carcinoma, and Blake helped us out with that as well. And then a number of other groups. This is our original ovarian work, and then another. It's become a bit of an industry in terms of what people have looked at. A lot of different solid tumors, and a lot of different groups. Interestingly, Karen Jerström's group in Sweden, in Lund, had actually noted that it was in buds, in invasive buds in colorectal carcinoma. And so that's why we got together with David Schaefer and had a look at some pancreatic, some PDACs, and we see the similar thing in buds. So this is just to show you in these little clusters, right? For the pathologists, coming off the main primary tumor, you have these little invasive buds. And the interesting thing in our minds at the time was, because experimentally thought we were seeing similar things, is that when you overexpress podocalyxin in tumor cells, you can make them invasive, but they're not mesenchymally invasive in that they're not single cells driving through the matrix. Often they'll move in clusters, and I'll show you some of that in a second. So this was the group in Sweden, and we see similar things. These are pancreatic buds, and the CK19 positive. They're really still quite epithelial, quite invasive, and they themselves are a prognostic marker of. And my lab really is interested in figuring out what would drive that kind of invasion. And I'll spend a few minutes talking about that in a moment, and then I'll come back to the clinical application now that we've made antibodies against this cell surface molecule. So I should tell you about Podocalyxin now, because I haven't told you anything about it so far. A lot of different folks have worked on this. A lot of cell biologists, so Marilyn Farquhar, so a lot of work in spheroid cultures of MDCK cells, these dog kidney cells that make nice balls, and Marilyn was working on it in those cells through the 80s and 90s. It was once called GP135. Ira Melman worked on it because it's excluded from cilia, and it seems to be important in setting up a domain where there's no podocalyxin that allows cilia to form on the apical domains of cells. Keith Mostoff, another cell biologist, showed that it was important for lumen formation when it's on the apical domain. And our collaborator, Kelly McNagny, who came at it from the point of view of an immunologist, because it turns out it's on a lot of immune stem cells. And actually, Kelly was the first person to actually clone it and make the first knockout. So it's a single pass transmembrane domain. Turn it on its side here. Normally, it gets transported to the apical domain, almost invariably above the tight junctions. But it will actually go to a non-adhesive domain even without junctions. So it's got some interesting targeting that if anybody wants to ask me about, I can talk about at the end. It's got a stubby cytoplasmic domain, a few little phosphorylation sites. It's got a C-terminal sequence DTHL, and this is a PDZ binding domain motif. And the most prominent PDZ containing scaffolding protein that it binds to is something called NRF1 and sometimes NRF2. And it also, that can interact with ezrin, and these are both can interact with the actin cytoskeleton. So that C-terminal domain interacts with the actin cytoskeleton. It turns out it can also, the NRF itself can scaffold with other receptors. So PDGF receptor, for example, EGF receptor, and can affect signaling. So a lot of folks are working on that. We're working on how it interacts with the cytoskeleton and the functional aspects. So I'm going to talk a little bit about that in a second, but I should tell you about the extracellular domain. So the extracellular domain, as you get out towards the end terminus, the end terminal 2/3, is highly glycosylated. 76 O-glycosylation sites, and you're seeing those with the little triangles on the end because most of them are silated, so lots of negative charge. And there's a few N-glycosylations sprinkled in there as well. And our first paper that we were publishing, and we were doing some functional stuff, and we made some tentative conclusions about what the extracellular domain was doing. And the reviewer, who I'm convinced was Marilyn Farquhar, asked us to do scanning mutagenesis on all the O-glycosylation sites, which we chose not to do, and sent the manuscript to another paper, to another journal. So N-O-link glycosylation, and a lot of this is silated, a lot of this is sulfated. So it's a major glomerular anion because it's in the glomeruli of the kidney, and that's how it gets its name because it's expressed on podocytes. And it's required for slit diaphragm formation. And that's really, Kelly's lab showed that. He made the original knockout. And when they knocked out the podocalyxin, what happens is that the podocytes, which normally push apart a little bit to get the slit diaphragm, essentially the one portion of the urinary filter, that doesn't happen. The cells stay together and stay tight. And I heard him give a talk once, and we didn't know each other. And I thought, that's an interesting molecule. If you forced express it on tumor cells, would it push them apart and allow them to break off from tumors and become invasive? And that was our whole original hypothesis, that we were going to mess up junctions and mesenchymally transform the cells. Never happened. I forced people in my lab and we tried really hard. Never happened. You'll see stuff in the literature where it says it happens. We don't believe it. It can influence the epithelial phenotype and perhaps drive a partial transformation, but not a full transformation. It's apically targeted. As I mentioned, it's excluded from the primary cilium, and that's really because of its interaction with this PDZ domain binding protein, nerfin, and then the esrin. And as I mentioned, it's usually tons of it on the podocytes, and you need it to push the cells apart a little bit to form the slit diaphragms. It's on a lot of post-capillary vascular endothelia and some other endothelia as well. There it seems to be important in lumen formation, and Kelly was involved in a study to show that. And interestingly, it's on the mesothelium also, which works up, I'm sure, lots of female reproductive gynecologic thinking folks there. The other knockout phenotype is you actually get an homophyseal where you get this midgut extrusion. And we think that's because the mesothelium, when it doesn't have this, is not as slippery, because this thing is really anti-adhesive through its extracellular domain because of all that charge, much like many other mucins. So that's the mouse knockout. So that's the molecule itself. And what we were doing in the lab is, in addition to what folks had been seeing with the clinical tumor buds, is that we found that when we overexpressed it, we would get a collective migration phenotype. So cells would move as a collective, both in vitro and in vivo, essentially mimicking the bud. So this is a classic, just a scratch assay that you see many cell biologists doing. So what's happened here is a monolayer has been scratched, and all this dark area in the center is where the cells have been removed. And then you look to see how many and how fast the cells migrate into this dark space. And what's happening as you move up is that you're going through time. And when we go from no podocalyxin, and this is MCF7 mammary carcinoma cells, quite epithelial, luminal actually, overexpressing the podo, you can see it's filling in the space really quite rapidly. And you can block this by pharmacologically inhibiting esrin or removing the cytoplasmic domain, that cytoplasmic tail of the podocalyxin. So it's all that interaction with the cytoskeleton that helps drive this. The interesting thing about this is that when it happens, when this migration happens, this is at the front. When the migration happens, it's not, the cells aren't releasing. And you can see the actin stained in green there. The podo is just in the red. You can see that the cells move as a collective. So this is a collective type migration in 2 dimensions, and you can model it in 3 dimensions. So these are the same cells now that would normally form spheres in a 3-dimensional context, but when you overexpress podocalyxin, they form these elongated shmoos that you can measure just by looking at the long versus the short axis. And this is due to contractility of the actin myosin-based cytoskeleton downstream of esrin. So if you block the contractility, you block this elongation, which is actually collective invasion, as I'll show you in a movie in a moment. And really what's interesting about what happens here is you get all these microlumens form as you get these protrusive edges that push out into the matrix. And recruiting esrin, which is just what you're showing here on the left, without the forced podocalyxin overexpression, and then the right with the podo. You can see the colocalization of the esrin in green and the podo. And here's what they look like. These are these little shmoos now that we are looking at in real time. There's a good one down here that's just pushing out. It's trying to let go. And this is a cluster of cells, and it can't do that. And this is that elongation, which you can quantify. Aaron in the lab is working again with the cytoplasmic tail to see really what interactions are critical to help drive this. and generate the contractility. Downstream of the receptor, there's a GTPase activation as well. We can also do it in vivo. So this is what you're looking at on the left here is an MCF7 tumor, orthotopic. So these are mammary tumors that have grown up in the mammary fat pad. This is the lymph node over on the left. And you get a nice smooth tumor sitting essentially in the fat of the fat pad, because these are very cohesive cells. And when we overexpress the Podocalyxin, what you do is we essentially drive tumor buds. So quite similar to what I showed you in those clinical PDAX and colorectal tumors. So we can drive selective invasion. So what about then if it drives the invasion? Does it have any role in experimental metastasis? And this is work that was done by Kim Snyder in Kelly's lab. So now we're going to switch breast tumor lines, talking breast here. We'll kind of flip-flop between tumor indications. But these are MDAM231 cells, which are highly metastatic, and you can put them in various places. You can put them into the vascular system and get them to go different places, but you can actually make primary tumors and then look and where they go. And Kelly, because in his lab, he thinks like an immunologist, rather than doing it like many of us would be stick in the cells with or without the gene of interest upregulated or downregulated, and then look to see individually what happens. He actually likes to put both cell types in and do a competition experiment, which is what they did here. So these MDA231 cells, which are highly invasive, also express lots of podocalyxin. So this experiment is a loss of function experiment as opposed to the gain of function experiment we showed a moment ago. And what they're going to do is they're going to fluorescently tag either wild type cells, the control cells, or cells where podocalyxin has been knocked down with a different fluorescent tag, put them in the mice, and then look to see the ratios in the metastatic situation. I'm going to show you data from the. So across the top, what you're looking at is on the left is the situation where podocalyxin is knocked down and the cells are labeled in green versus the control cells. You put both into the animal. If there was no effect, you'd see equal ratios of the green and red cells. And the inhibitory effect in the podo knockdown, you see very few green cells get into the lungs where lots of the control cells have gotten into. And you can flip and do the opposite. On the right-hand side, the controls are in green, whereas the reds are the knockdowns, and now you see the ratios flipped. So that's distal metastasis that's come from a subcutaneous tumor, and you can see there's quite a difference in the tumor morphology as well. The knockdown, they're much less invasive than in the. So you actually get a metastatic. So this is where we changed gears and decided, made a big decision a while ago that's led us to rope in many of you to give us a hand, or always badgering you for clinical samples, which we very, very much thank you for, or your input on what we're seeing. It's been really great. Given that we had a protein that's expressed on the surface of lots of different tumor types, especially on high-grade serous ovarian carcinoma, and it looks to be functionally important in progression, and many other people are doing functional experiments as well. We decided to try and make antibodies to see if we could make some kind of way to modulate function by binding to the extracellular domain. And to do that, we engaged the help of those folks I mentioned right at the beginning, particularly John Babcock, and then Chris Bond and Ishmael Samudio came and helped us afterwards. But it was really John's SLAM antibody technology that he brought to CDRD from Amgen here in Vancouver, and really developed when he was working with John Schrader and part of that group in the Biomedical Research Center, the Wellcome Burroughs group, back a number of years ago. But John made us a whole bunch of antibodies against podocalyxin that was expressed on the surface of those highly metastatic tumor cells that I just talked about, those MDA230. And we tried to develop an assay that would tell us which ones might be functionally important. So they're binding to the extracellular domain. The idea is, would we interrupt that kind of mucin anti-adhesive function? And the screen that we developed was that it turns out when cells are highly expressing podocalyxin, they stick and spread on a rigid surface quite slowly. So the idea was if you block their function, they'd stick and spread more rapidly. And we found a whole bunch of antibodies that would do that, but we had really low discriminating power. So Kelly and Kim Snyder in his lab took the leap, coated a whole bunch of cells with antibodies, put them right into animals and did the xenografts, and then looked to see if any antibodies would inhibit the growth of those xenografts. And one of these antibodies, we screened a number of them, but one of them was the antibody that we've used a lot, and this is Podocalyxin 83. It's just a clone. And you can see it, it's the one in the black triangles. Over time, tumor volume in vivo, it just essentially knocks it right down. We had some variability in some other antibodies, but this was really the best one. And what's important to realize here, this is a naked antibody. So this is naked antibody inhibiting primary tumor growth, and in that same metastasis assay, inhibiting metastasis to lung. So it's an interesting functional antibody. The issue, though, is that this antibody, Podocalyxin 83, it binds to the stalk domain on the extracellular portion of human Podocalyxin, doesn't recognize mouse Podocalyxin, human Podocalyxin on tumors and on kidney and on the vasculature and on the mesothelium and probably on some other tissues that I'll tell you about in a minute. So the question is whether this antibody can ever be used therapeutically. One of the ways we're working to assess that right now is that we've made a human podo extracellular domain knock-in mouse. So we're going to be testing function there as well. So whether we'll be able to move on with Podocalyxin 83, this antibody, we're not sure. But we have another antibody that doesn't work as a naked antibody, but is an interesting one that many of you will have heard of. And we're trying to change the nomenclature. We're trying to call it now, at least I am, I don't know about Kelly, but I'm trying to call it T-Podo, because I know for Yemen and David and Blake, it drives them crazy. T-Podo and Don and Spencer, we're trying to call it T-Podo because it binds to what we think is a pretty tumor specific, or at least tumor restricted epitope, glycoepitope on Podocalyxin, out in the N-terminus. And we think it's a modification, and I'll tell you about that in a little bit in a moment, in the O-glycosylation of the extracellular domain of Podocalyxin with the Podocalyxin backbone. So we think it binds a peptidoglycan epitope, mostly on tumor cells, and not on most normal cells. And the idea here is we're now been working to make an antibody drug. Okay, so that's T Podo 447, or often we call it 447. So this is just to show you that it doesn't bind normal tissue. So on the left, what you're looking at is kidney, and this is that Podo 83 that binds everything on the left. On the right, this is staining kidney with the Podo 447, and affecting kidney function was our biggest worry. So the 447 is not binding human. Most other tissues that we've looked at, we really see very little binding. Sometimes we do see a hint of vascular binding, particularly the tumor vasculature. So we don't know if that's a change that's happening in tumor vasculature, which we're interested in following up on. As well, Lynn and Spencer Martin in particular have really, and working with Don, there is a caveat here though, that now that we're looking hard, working with the group, we are seeing some staining in normal endometrium, from the endometrial glandular epithelia. And looks like the secretory cells, although it's somewhat patchy, the oviductal epithelium. Now working with Spencer and Don, we're trying to figure out how that's regulated and why it's coming up. But to do that, one would like to know what changes the glycosylation, or what the changes are, and perhaps some of the glycotransferases that might be affected to regulate the change that allows 447 to bind to tumor, but mostly not normal cells. Oh, I haven't actually shown you that 447 still binds to tumor. So important to point out that it doesn't bind as widely on tumors as the 83 does, as you would bind all of the Podocalyxin. So it's a pretty significant subset. Now we're going back to high-grade serous tumors again, and this is work that Sam helped us out with, as did Martin. If you remember, we were looking at 90%, and in fact, with our new antibodies, it's actually higher with the antibody that sees all podo, but the podo that sees the tumor form, this 447 antibody, it's about 2/3. And there's quite a significant proportion that's on the cell surface. And it's also within other ovarian types as well. An interesting correlation that this is the thing, and just a little bit of tantalizing bit of data, and that Julianne's working hard on, and we're hoping to look in the endometrial situation as well, is that Julianne, working with samples from Brad Nelson's lab in Victoria, where they've looked at a lot of infiltrating cells, immune cells. And in this case, we're just showing you the CD8, CD4 T cells as well as B cells. What you're looking at on the y-axis is, the percentage of infiltrating lymphocytes. And on the x-axis is the amount of podocalyxin. So the percentage of cells staining with podocalyxin as you move out to the right, you see quite a drop in the immune cells, in the immune infiltrate. So it seems like there's an inverse correlation between the appearance of this epitope, this funky epitope on the surface of the tumor cells, and decreased lymphocytic infiltration, also some innate cells as well. Why that's happening, is that really due to the 447 tumor cells themselves, something in the microenvironment that gives rise to that 447 epitope? That we're not sure of, and that we're trying to work out experimentally. And that's part of Julianne's project in Kelly's lab. This is just a summary slide just to show you, first of all, it's not a fluke in the situation, in the ovarian situation. Not only have we looked at the GPEC array, we've also looked at the array from Anne-Marie Mesmasson in Montreal, the CHUM array. Number of other, we've had a look at urothelial pancreas endometrial, and that's really, Navina did that original work, and why we're working with Yemen on project 3 of the TFRI grant, breast as well. And what you're seeing on the left is the 83 that sees everything, and then the 447 is on the right, and you can see that you drop down because it's a subset. And we're looking at some of these other ones in the bottom as well. So we think it might have some utility in other tumor types also. All right. So what's it actually binding? What is that 447 antibody recognizing that has this tumor restriction and is not seeing all the podocalyxin? So to try and work this out, this is where we engage in the collaboration with the fellow and a student in Copenhagen, Ola Blixt and Iliara. And Ola has a really interesting system. Essentially what he has is HEK293 cells, so human embryonic kidney cells that everybody uses for just about everything. Easy to get things in, easy to get things out. They themselves actually express podocalyxin. And then what he's got is cell lines where he's knocked out all the glycotransferases that are important in O and N glycosylation. So what we're looking at here are things that will manipulate what happens in O glycosylation. And as he messes around with various transferases, we're looking to see how much the 447 antibody binds to the surface of these cells. So in the spider web, when you move out to the outer aspects of the spider web, you're binding more. If you're in at the center, you're not binding at all. So it's just a way to show the accumulated data with the various ways to manipulate the cells. So all these manipulations that give rise to funky truncated bits of glycosylation, for example, the TN antigens, and this one you just have N-acetyl-galactosamine and then nothing else happens because the transferase isn't there to allow things to get added onto, doesn't bind 447. So there's no binding in this spoke of the spider web. And that was an important one for us to look at because TN antigen is truncated glycosylation that happens on a lot of mucins, including MUC1. And this is something that other folks are targeting. So Carl June's group, for example, is targeting this in solid tumors, in fact, with CAR-T-based methods to actually try and find the tumor cells that have this truncation. This is a variation on the theme. This is one just where it's silated, where it can be silated. This is a funky core 3 one. But when you come around to this side, where you're getting these little doublets either without silation or even with double silation. Now we're starting to see 447 binding. So what you have here is you have the original N-acetyl galactosamine, and then you have galactose stuck on the surface. And when you have that, that's when you have the highs. And this is a different carbohydrate. It's not the same as sp40 T antigen, but for these glycobiology folks, they call it the T antigen on O-linked sugars when you have this stubby little guy. So either without or with the silation, and even better without the silation, we get good binding. So you could ask yourself, is it just binding that sugar? And if that sugar, if that little stubby sugar showed up on any O-glycosylated mucin, would we have binding? We don't think that's the case. We think that the 447 antibody recognizes this little stubby glycosylation in the situation where the podocalyxin protein backbone is there out in the N-terminus. And one of the reasons we think that is based on this experiment. So these are SKOV3 cells. SKOV3 cells in ovarian carcinoma line has tons of, how am I doing for time? I mean, I'm not running late. Am I? Oh, good. I'm almost good. SKOV3 cells have tons of podocalyxin. So if you stain them on the, just flow cytometry on the x-axis, these are just the controls up here. These are the wild type SKOV3s. And then this is staining with 447, the tumor specific form. So it recognizes it on SKOV3 cells. Or if you use the other antibody that sees everything, it also binds. And then if we knock it out, we get no binding. What you'd expect to get rid of the podocalyxin. If we stain these same cells with an antibody against the TN antigen, there's still binding of the TN antigen. So even though once we take the podo away, we don't see any binding of 447, even though there's lots of TN antigen on these cells. We really don't think that this antibody is just seeing TN antigen, it's seeing TN antigen in the context of, or the other things, because there are other podocalyxin-like molecules in the CD34 family that Kelly works on that I can tell you about if anybody wants to ask about. But we haven't seen it yet. So that's what we think it's binding. And we don't think it's binding to most normal tissues. So we think we can maybe make an antibody drug conjugate, which we've also done some labeling with Francois Bernard. We've taken some baby steps towards making a CAR T, but that hasn't moved so far along the road yet. So I'm going to tell you about the antibody drug conjugate. And as I mentioned, a lot of this work was done by Diana Canales-Hernandez when she was in Kelly's lab, together with Pam Dean. Well, this is just a summary. This is a really nice little drawing that Julianne made recently for review. So we have a nice review on this if anyone's interested. This is just kind of to show you the way we think about it. So here's podo, and this would be the podo that 83 could bind to down here in the stalk in a normal cell. And this is how we're thinking about it in the tumor cells where we're getting these truncated bits. And we think the antibody is binding both in the context of the peptide backbone and this little stubby bit, either silated or not, that's hanging on. So that's kind of the idea. What gives rise to it, as I mentioned, is we're looking into these glycotransferases that were manipulated by the Blixt group in Copenhagen. And of course, the other thing we've done is that Aaron and Pam in my lab, we have all different cell lines, some of them that have populations that have lots of podocalyxin, some high 447, some low 447, and we've separated those and then done RNA-seq. We see some of the transferases and we're working on those. But the other thing we found is we see another interesting aspect of cells that have high 447 is that we see a lot of genes that would come up normally on an EMT signature. So for example, CDH2 is NCAD-herin, Vimentin is the mesenchymal intermediate filament gene. And these are a number of the regulatory genes that can influence cell-cell adhesion gene expression, and then also things that interact with the matrix. So we see these, and Aaron's looked really hard because I said, voila, finally, the hypothesis we had 20 years ago is coming true. We're going to mesenchymally transform these guys when we have high 447, and it turns out that's not the case. We really think it's the partial EMT. These things still migrate and invade collectively. I just think perhaps more so. Whether this phenotype is associated with any kind of a stem phenotype, That's something that we're working on at the moment. Okay, finally, closing act, finally getting to the ADC. So we've got the T Podo 447, and what we're going to hang off of the Podo 447 with a protease cleavable linker, meaning that once you get it into cells and get into the endosomal lysosomal pathway, it can be cleaved, and it will release this MMAE or betatin. So this is the actual armature. This is it's actually a microtubule poison. And the construction of this ADC is quite similar with the protease cleavable linker, and the MMAE is quite similar to the recent FDA approved drug that targets the folate 2 receptor in ovarian tumor cells. So it's kind of a similar theme that we've used. Okay. It gets taken up by cells. So this is, we've just made it fluorescent, we put a fluorescent tag on and it's getting taken up. We're a control, we use a control here that actually is against, I'd forgotten about this given where I am today at the hospital against one of the fusion proteins in RSV. But it doesn't, that one doesn't get taken up because there's no receptor, that molecule is not on the surface of these tumors. It is cytotoxic in vitro. So going back to the SKOV3 cells that I told you about before, the blue is a control antibody drug conjugate. So this is the one that doesn't bind to the surface of the cells. And then this is the one that binds the tumor Podo 447, and that this is the cytotoxicity in vitro. If we knock out the Podo, we don't have that in the SKOV3s. And this is the same in a pancreatic line called Miopaca, pancreatic carcinoma line, the same thing. So in the wild type, we get nice killing. If we knock out the Podo, we don't have killing. This more real world, at least in the world of cell lines, these are 2 patient-derived high-grade serous ovarian lines from Anne-Marie Mess-Masson that have Podo 447 on their surface. We haven't done the knockouts yet, but this is the situation. We get nice killing that's, again, 447 ADC specific as opposed to the control. It's 2 different. So it has some nice utility in vitro. And now we're doing the kinds of experiments in vivo, and these are the kinds of experiments we hope to be doing with the endometrial group as well, and the TFRI grant, where we've done things in vitro with xenografts. So rather than just showing you the survival curves, we just wanted to show you all the data. And so that's what you're seeing. This is all the each dots from different tumors over time on the x-axis that have received infusions of the ADC early on in the treatment. And early on in the treatment, we get a really nice delay. In the red is when they're getting the 447 directed ADC. And in the blue is the control, where things are coming up much faster. And therefore, the survival curves drop off in those guys. And that's with the Miopacas. And this is one of Anne-Marie's lines, the high-grade patient-derived lines, and not quite as good a response. But we get a significant delay, and now we're tweaking and looking at adding various other compounds to see if we can really get a nice, robust response. So that's it. I'm all done. Thanks so much for everyone that's given us a hand. I know many of you are on the call over the various amounts of time. Kelly and I were talking about this the other day, how many folks we've roped into giving us a hand on this. So thank you very, very much. This is one of those spheres with normal cells with Podo in red on the apical domain. The green is Z1 per tight junctions. And this is back to Julie's original stuff and Marcia's when they're in the lab. When you overexpress Podo, you get these bulging apical lumens. So in a context of normal polarity, you can actually have, we think, also ductal infiltration.